This is the first of a two-part series of videos that are meant to show you how to utilize a device called a hemocytometer in order to count the number of cells present in a sample from a cell culture. So in this first video, I'm simply going to demonstrate how you add cells to the hemocytometer, and then in the second installment, I will demonstrate how you use a microscope to count the cells that are present on the hemocytometer. So, what is a hemocytometer? A hemocytometer is basically a specialized piece of glassware, kind of like a microscope slide, except it has two kind of very highly polished and translucent surfaces on it. These are referred to as the counting chambers, and they are the places where we add our cell cultures. Now, the adding of cell cultures is a little bit different than you would do if you were making an ordinary wet mount slide. You use a cover slip, like you can see here, but rather than adding the liquid onto the hemocytometer and then placing the cover slip on top, we actually put the cover slip on first. And we do it and slide so that the bottom edge of the cover slip goes a little bit past the lower counting chamber. Now you might be able to see that there is a small beveled surface or a notch located right at this point. This is where we're actually going to place our liquid and allow it to be drawn up through capillary action underneath the cover slip. We don't need a lot of liquid. In fact, for this experiment, we only need to add 10 microliters. So we will be using a micropipetter that should be set to 10.0 microliters. So we've already added a micropipetter tip onto it. Now all we need are our cells. Our cells should be present in a small rectangular container from the previous lab activity, and this container is known as a cuvette. So all we need to do is get a little bit of a sample and add it to the hemocytometer. So go ahead and take your pipetter. Remember to press down to the first soft stop to prime it to take 10 microliters. Put the tip in the cuvette. Let go of the button. You're going to draw it up. Now to add your liquid to the hemocytometer, what you need to do is that you need to bring the tip of your pipetter into the front edge of that notch just underneath the cover slip, and then begin to slowly press and get the liquid to go up underneath. Take it slow and steady. And there you go. Now, for kind of efficiency in your setup, you do need to actually add another 10 microliters to the other counting chamber. So that's going to require you to turn the hemocytometer around. You can somewhat slide the cover slip over to the front of the other notch, and then you can use your same micropipetter, grab 10 microliters, and again, you need to go in. Notice that it's really important, one, to hold with both hands so you have a steady hold, and that you're kind of coming in sideways with the micropipetter as you're adding your liquid. Once you've added 10 microliters to each side of the hemocytometer, now you're ready to place it onto a microscope and begin to focus. And that we will demonstrate in the next video.